Today I will be talking about DNA replication. DNA replication is one of the most important processes in the body. Before we get through the process of DNA replication, what even is it? DNA replication is the process of DNA making a copy of itself during cell division. So, this means that during processes like meiosis or mitosis, the cell splits itself in half in order to make copies for each. But why do we have DNA replication? DNA replication ensures us that each cell contains a copy of these instructions, or DNA, and is able to function correctly within the organism. So here you see a picture of DNA and its complementary base pairings. Note that A pairs with T and C pairs with G. The first step of DNA replication is to unzip the DNA. This is done by an enzyme called helicase. Helicase is able to unwind the double helix by breaking the hydrogen bonds between the complementary base strands. So basically the bonds between A and T and C and G are broken by helicase in order to create the unwounded DNA. Note, just like Os equals sugar, such as glucose, like lactose or sucrose, Ase equals enzyme, such as helicase, glycase, or primase. Once the DNA has been unzipped, the Y shape is called the replication fork. Even though it doesn't really look like a fork, because it would have to have a red line to it to actually seem similar to a fork, this shape serves as a template for making DNA. The leading strand and lagging strand. Once the DNA is shaped into the replication fork, DNA orients itself into the leading strand and lagging strand. The way the arrows point, either towards DNA or away from DNA, is the way that they will make their base pairings with the other parts of the DNA. So here the 5 to 3 strand is a leading strand, and the leading strand will always move toward the replication fork. The 3 to 5 strand is the lagging strand, and will always move away from the replication fork. Note that the lagging strand has the Okazaki fragments, which is why there are 2 arrows instead of 1. Also note that leading strand and lagging strand have different ways of replicating. So first we'll be talking about how the leading strand replicates DNA. So firstly, the primer comes to the end of the leading strand to start the replication process. You wouldn't be able to replicate DNA if it wasn't for the primer, and the primer comes from the enzyme primase. The DNA polymerase binds onto the leading strand and walks across it, making complementary bases for the new DNA. So basically the DNA polymerase starts from where the primer starts off and continues to move through the DNA in order to make the base pairs that it needs to replicate. It moves towards the replication fork in order to replicate all the DNA. Okay, so next is the lagging strand. So the primer starts on the opposite end and continues to make fragments for primase. So basically the primer starts in the middle of the DNA fragment and moves toward the end like the way it's supposed to replicate. The primer makes a separate extended part for the new strand of DNA and all the Okazaki fragments. So basically the primer makes the extended part of the Okazaki fragment in order to keep the complementary base pairings continuing. The Okazaki fragments are short segments of DNA, and once DNA replication is finished, all the Okazaki fragments join together to be one strand of DNA. So once all the bases in DNA have a complementary pair, the enzyme exonuclease strips away the primer. So basically it's taking away anything that it doesn't need anymore. And the gaps that are left that don't have any bases are filled with the nucleotides, which are A, T, C, and G. Next, the newly made DNA proofreads all of its pairings and makes sure of any mistakes. So to make sure there's no mistakes in the DNA replication. So during the final steps of DNA replication, the result of DNA replication is 2 DNA molecules that contain 1 old strand of DNA and 1 new strand of DNA. This is called being semi-conservative, where it has 1 old and 1 new. After that, DNA automatically winds up itself from the replication fork and turns into the double helix structure of DNA. Thank you for listening to my presentation on DNA replication.